Good day viewers. Today in this video I am going to discuss about lysosomes. Now let's see about the introduction of lysosomes. Lysosomes are membrane bound organelles found mainly in animal cells. Discovered by Christian D. Dewey in 1955. They are known as the suicidal bags of the cell because they contain large number of powerful digestive enzymes. These enzymes can destroy cellular components if released into the cytoplasm. Lysosomes are usually spherical in shape and about 0.1 to 1.2 micrometer in size. They mainly function in intracellular digestion, defense, and recycling of cellular materials. Now we'll see about the structure of lysosomes. Lysosomes is surrounded by a single lipoprotein membrane. This membrane is very important because it separates the powerful digestive enzymes inside the lysosome from the rest of the cell, thereby preventing damage. The membrane contains special proteins such as lysosome associated membrane proteins, which are heavily glycosylated and protect the membrane from being broken down by its own enzymes. It also contains transport proteins that allow digested materials like amino acids and sugars to move into the cytoplasm for reuse. The lysosomal membrane also contains proton pumps that maintains an acidic environment inside the lysosome. The inner space of the lysosome is called the matrix or lumen. It is filled with an acidic fluid that contains more than 50 different types of hydrolytic enzymes. These enzymes include protease, nucleases, lipase, glycosidases, phosphatases, and sulfatases. These enzymes are capable of breaking down proteins, nucleic acids, lipids, and carbohydrates. The acidic pH around 4.5 to 5 inside the lumen is essential for the proper functioning of these enzymes, and this pH is maintained by proton pumps present in the lysosomal membrane. The lysosomal membrane contains a special enzyme called vacuolar type proton pump ATPase, which acts as a proton pump. This pump uses energy from ATP to move hydrogen ions from the cytoplasm into the lysosome. As a result, the lysosome becomes highly acidic. This acidic environment is required for activating the digestive enzymes and allowing efficient breakdown of cellular materials. Without this proton pump, the enzyme is inside the lysosomes would not function properly. Formation of lysosomes. Lysosomal enzymes are synthesized by the ribosomes on the rough endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus where they are modified and packed into small vesicles. These vesicles bud off from the Golgi and form primary lysosomes. When a primary lysosome fuses with a vesicle containing food particles or old organelles, it becomes a secondary lysosome where digestion occurs. In this way, lysosomes are continuously formed as a part of the cellular transport and recycling system. Then types of lysosomes. Primary lysosomes are newly formed and contain digestive enzymes, but they have not yet participated in digestion. Secondary lysosomes are formed when primary lysosomes fuse with phagosomes, pinocytic vesicles, or autophagosomes, and these are the sites where active digestion takes place. After digestion is completed, undigested waste materials remains in a structure called residual body. These residual bodies may be expelled from the cell or may remain inside the cell for a long time. Now we'll move to the functions of lysosomes. The main functions of lysosome is intracellular digestion. They break down substances such as bacteria, viruses, damaged organelles, and macromolecules like proteins, lipids, carbohydrates, and nucleic acids. When a foreign particle enters the cell through phagocytosis, the phagosome fuses with the lysosome to form a phagolysosome. Inside the structure, enzyme digests the particles into simpler substances that can be absorbed and used by the cell. Autophagy means self-eating. In this process, lysosome digests old, damaged, or non-functional cell organelles such as mitochondria and ribosomes. This helps the cell to get rid of worn out components and maintain healthy functioning. The broken down products are recycled and used for the formation of new cell components. Thus, autophagy plays an important role in maintaining cellular balance. And the next function is defense mechanism. In immune cells such as macrophages and neutrophils, lysosomes destroy harmful microorganisms. After these cells engulf bacteria or viruses, lysosome fuse with phagosome and release digestive enzymes that kill and digest the pathogens. In this way, lysosomes help protect the body from infections and play an important role in the immune response. Then autolysis. If a cell is seriously damaged or infected, the lysosomal membrane may rupture, releasing digestive enzymes into the cytoplasm. These enzymes then digest the entire cell, leading to a process called autolysis. This is why lysosomes are called the suicidal bags of the cell. Although this leads to the destruction of the cell, it is beneficial for the organism because it removes damaged or dangerous cells. Then recycling of materials. After digestion, the simple substances like amino acids, fatty acids and sugars are transported into the cytoplasm. These substances are reused by the cell for energy production, repair and synthesis of new molecules. In this way, lysosomes act as the recycling center of the cell, ensuring that useful materials are not wasted. Now last we'll see the clinical importance of lysosomes. If any lysosomal enzyme is absent or defective, undigested materials accumulates inside the lysosome, leading to a group of disorders known as lysosomal storage disease. Examples are Tay-Sachs disease, Gaucher's disease, Pompe's disease, Hurler syndrome, etc. These conditions cause damaged tissues and organs, especially the brain, liver, and bones, and may lead to severe physical and mental problems. Thank you for listening and watching the video. Have a good day.